Hi everyone, today we'll be talking about transcription and gene regulators as well as kind of the behind the scenes networks that regulate transcription. So a brief overview of transcription. It's part of the central dogma and it converts, or it's an intermediate pathway in order to convert DNA to protein. So we mainly focus on mRNA transcription. This is done through RNA polymerase II, but transcription occurs in all types of RNAs. So transcription is a term for RNA synthesis from DNA. This includes ribosomal RNA, messenger RNA, tRNA, and microRNA. So a few ways that transcription differs from replication. Uracil is a nucleotide used in the place of thymine. Ribose sugar is in an RNA molecule instead of deoxyribose, and there's no proofreading activity. This is done in replication through DNA polymerase I, which has 3' to 5' exonuclease activity, but RNA polymerase only has one polymerase enzyme that goes and synthesizes from 5' to 3'. So there are 3 steps to transcription, and the first is initiation. This involves many proteins, which form protein complexes involved in initiation, eventually creating the transcription initiating complex. So a general class of transcription factors are the general transcription factors, and these are necessary for transcription to occur. However, with only these factors in RNA polymerase, the rate of transcription is really low. So to increase that, we have specific transcription factors that bind to control elements. And control elements are non-coding DNA strands that are upstream of the target gene that's to be sequenced. And really far control elements are called enhancers. These specific transcription factors will greatly increase the rate and binding affinity of RNA polymerase. A third class of proteins are the activators and also the activator DNA sequences. These proteins attach to the DNA sequences and they also recruit the DNA bending protein, which allows proteins that attach to control elements to go with proteins, the general transcription factors, and this in turn creates the transcription initiation complex seen here. With this complex, we can then have RNA polymerase bind to the promoter of the gene that we're trying to transcribe. So the second step is elongation, and this is where the RNA molecule is actually synthesized. So RNA polymerase starts at the promoter and synthesizes RNA in the 5' to 3' direction, and it detaches when it reaches a certain termination sequence. And the transcript produced by this is called the primary transcript, or pre-RNA, and this is the RNA before any modifications. So the third step of transcription is modification, or post-transcriptional modification, and this comes in 3 main categories. For 5' modifications, we have a guanine or a modified guanine cap at the 5' end, and this allows the mRNA, in this case, to bind to the ribosome for translation. On the 3' end, we have polyadenylation of the 3' tail, and this is basically adding a lot of adenine nucleotides to the end of the 3' tail. And this protects the mRNA from being cleaved by exonucleases and other RNases in the cellular matrix. The third type of modification is introns and exons, and intron splicing in eukaryotes allows for different combinations of different exons, and this produces a lot of different protein sequences that can occur from the same DNA strand. This is called alternative splicing, and it really increases the amount of proteins that we're able to have from a smaller amount of genes. However, this isn't present in prokaryotes because prokaryotes don't have a nucleus. So the final thing that I want to talk about is transcription networks. And we are only beginning to research how transcription is really regulated. So being part of the central dogma pathway, transcription regulation is a really crucial part of gene regulation and expression. And organisms have evolved to form really complex transcription networks. So the picture below is the E. coli transcription factor network. And here it's represented in a graph with vertices or nodes at the dots and edges connecting the genes. So the green dots are transcription factor genes, and the brown dots are regulated genes. So you can really see the complexity of transcription regulation. And as we research more, this is a really key topic in computational and systems biology. And a key part of this is finding network motifs, so different subgraphs of the main graph, and seeing how they modify or change transcription regulation. So this has been a brief summary of transcription and as well as transcription networks, and I hope this was helpful.